Fine. Thank you. Yeah. Okay, shall we start?
Yes.
Yeah.
So should I share the slide?
Sir, I'll just give me a few minutes. I'll just give
Okay.
it. Yeah. Good morning, everyone. I welcome you all for this special talk, which is given by Professor Manoj Prasad. So, Professor Prasad is currently a professor and senior scientist at National Institute of Plant Genome Research, New Delhi, India. And see, Professor Prasad is done all his, higher education from University of Calcutta, including BSc, MSc, and PhD in botany. And his research interests are like plant molecular genetics, genomics, and stress biology and biotechnology, and biotechnology for to improve crop, both nutritional and also the disease resistance. And he has been doing an excellent work since he joined NIPGR, and he has published more than 200 research papers, including reviews, like popular articles in many of the internationally reputed journals. To name a few, the PNAS, he recently got a PNAS paper, and Plant Journal, Journal of Experimental Botany, DNA Research, and Theoretical and Applied Genetics. And he also got excellent reviews in various trends journals like Trends in Plant Science, Genetics, and Microbiology, etc. And he has got a very good citations as well, and he has got more than 13,000 citations and H index of above 65. I mean, all this only says that how active he is in research, and how wonderful research he has been carrying out. And with this, like you can expect that definitely he has got numerous awards. To name a few, he's also a recipient of prestigious J.C. Bose National, is like J.C. Bose Fellowship, and he's also fellow of all the three national academies. And he's got Silver Jubilee Commemoration Medal and Tata Innovation Fellow, National Bioscience Award, Birbal Sahni Medal, and he was also like Alexander Van Humboldt Fellow, and Golden Peacock Award from Slovenia, and Hiralal Chakravarti Memorial Award from Indian Science Congress Association, and he also has got Young Scientist Award from NAS. So with this brief introduction, I would like to request Professor Prasad to start his talk. Sir.
Okay. Thank you, Shiram.
Yeah.
Let us see. Should I share the?
Yeah, you can share
the screen.
You can share the screen.
Shiram, you mute because
Okay.
echo is happening.
No, once he starts, then I will mute. Otherwise, he can't.
I have shared. Nothing is.
You can say ignore.
Ignore. Okay.
Yeah. And present full slide.
No, but where it is?
And your PowerPoint full slide.
PowerPoint slide. Yes.
Yeah. Yes. Perfect.
It came.
Yeah.
Okay.
Sorry, Professor Prasad, this is Shubhatim. There is an echo happening.
If everybody mute their mic, then I think there is no echo. Okay. So, good morning, everyone. I am audible? Sriram?
Yes, you are audible.
Okay.
Yes, go ahead.
So, please everyone mute your mic, then I will clearly audible. So, good morning, everyone. It is a nice meeting you all through this virtual platform on the occasion of special lecture. And my foremost thank to Dr. Tapasda, Supratim and Sriram for organizing this special lecture. And I also like to thank the students, especially, for joining me to listen my talk. So, as I said that I will talk about the Xanthomonas oryzae pathobar oryzae, a vascular pathogen of rice, how to control this bacterial leaf blight by genome engineering and other biotechnological tools. So this bacterial leaf blight was first reported in Japan by the Japanese farmer in Fukuoka area in 1884, were considerable to be caused by the physiological factor and acidic dew. So at that time, it was not known which is the causal organism and how this disease has been uh, coming. So later on in 1990, the actual causal organism was identified and named as Xanthomonas oryzae pathobar pathobar oryzae. This is the region it was known as zoo. Xanthomonas oryzae pathobar oryzae. So X stands for Xanthomonas. First O stands for oryzae, and the third O also stands for pathobar oryzae. Henceforth, the bacterial leaf blight occurs mainly in the Southeast Asia and the South African region. So wherever it is rice growing area, everywhere this bacteria leaf blight was occurred. So being a vascular pathogen, zoo interfaces were ranged from physiological to biochemical exchange process in rice. So I will discuss in detail about this, how this disease has been progressed. So this is the view of the field, and this is the closer view of infected plant. You can see here the bacterial leaf blight is in the margin of the leaf, and it is in brown in color. And this is the ooze, that infected leaf bacterial ooze that comes out of the through hydrathodes, and it is showing that the bacteria load is very high, and the ooze comes out as a overload of this bacterial infection, and how this bacterial culture looks like in a petri plate. So regarding this Xanthomonas strain, this is basically a gram-negative, non-spore forming rod-shaped bacterium, which is motile with a single polar flagellum. So you can see here single polar flagellum, and this is a rod-shaped structure, gram-positive. And so far as the cell size is concerned, 0.7 to 2 micromole in length, and basically 0.4 to 0.74 micromole in the width required. And optimal temperature for growing this is 25 to 30 degree for their growth. So in India, the red circle indicates the disease incidence in various locations across the Indian states. So almost all the rice growing area, it was devastated actually. So the disease incidence was reported long back in 1963 and 64 in Bihar first, and then 65, 66 to 2010, 13 in Andhra Pradesh, and then 80 to 81 in Punjab, Haryana, Western Uttar Pradesh, and 1998 in Kerala, and 2014 in Tamil Nadu. So in different years, this disease has been progressed throughout the rice growing areas. For the past, if you take the disease survey of 34 years, it was shown that the rice growing areas were highly infected with this bacterial leaf blight, and the yield loss causes up to 50% or more. So this is the disease cycle. As I said, that is a rod-shaped bacteria, and the flagellum is the signature of this pathogen, and it infects the rice sapling in the field, and it goes to the bacteria spread through this rain splashed or wind blown to cause the plant wounds, to infect the plant. And then how it looks like, you can see here the browning stuff, and this is the cross section of the leaf. You can see that inside the tissue how the zoo are infected outside the rod-shaped structure, and after the infection you can see the whole devastated field was brownish in color, and after the post harvest how it looks like. So everything has lost due to infection. So what plant used to do, once this bacteria infect, the plant has the cell wall, okay, to protect from this pathogen. But the bacteria needs to grow through this barrier to dissolve the cell wall of plant, so that it can get the access of nutrient from the plant to survive in the plant. And at that time, bacteria secretes some cell wall degrading enzymes, okay, to invade through this plant tissue, leaf tissue. Then the bacterial pathogen zoo gets into the leaf tissues generally via the wound or natural openings such as hydrothodes. What are hydrothodes? Hydrothodes are specialized spore on the leaves of the higher plant that function as a exucation of water. Through this is the hydrothodes. In the leaf tip margin, you can see here that special pore size structure. Through the pathogen infect. Subsequently, it multiplies and flourishes in the intercellular space, that is apoplast. Where is the apoplast? This is below the cell wall. You can see here this orange color, and the above the cytoplasm. This is above the beneath the epithelial cell actually. Then once it crosses this cell wall of the plant, it comes to the apoplastic region for its further growth. And thereafter, it disseminates the whole plant system by the xylem vessel. So it first degrades the plant cell wall and comes to this to the apoplast, and then from apoplast through this xylem vessel, it goes to all over the plant system. After a few days, the xylem vessels are filled with bacteria and its exopolysaccharide. The bacterial with its exo can be observed on the leaf surface as they come out through the hydrothodes. So this is the outcome of this, that through this spore hydrothodes, the bacteria will come out when it is accessed in the vessel, xylem vessel. So the first line of immunity, as I said, that the cell wall is the first line of immunity for any plant system, and the bacteria, the zoo effector has two things, that is the responsive regulator and the histidine kinase. So through this effector, they try to invade the cell wall, and the cell wall is made up of cuticle, and they are the passive barrier to the pathogen in rice plant, whereas active barrier encompasses secondary metabolites. So once the pathogen, the effector, cross this cell wall, and then the second line of defense, the secondary metabolites with antimicrobial activity, callose deposition, lignification of the cell wall, and cell death. They try to protect the rice plant from this invading of this pathogen. And what are the other characteristics of the cell wall? Cell wall is subject to reinforcement mechanism with superoxide production, and this step is accompanied by phytoalexin antibiotic compounds. You can see here phytoalexin antibiotic compounds, cell wall hardening things, and many other stuff. Cell wall architecture is importantly also defined by polyglucoronidase protein, which depolymerizes the pectin by hydrolysis and active cell wall immune response. So basically, the zoo try to invade the cell wall. Cell wall try to protect the host plant from this infection. Once the zoo cross the cell wall, then the secondary metabolites activates and try to protect the plant. The second line is the pattern recognize trigger immunity. So first line I have talked about cell wall. The second line of is the PAMP trigger immunity. So what is PAMP? PAMP is there is a pattern of this zoo from the bacterial pathogen, which is either salicylic acid or chitin or peptidoglycan as a molecular signature, which enable the plant to identify the invaders using the pattern recognition receptor on the plant cell surface and activate the plant defense response called as PTI. So this molecular signature pattern of this pathogen zoo, this yellow color you can see here, it was recognized by the host plant, the rice plant, some to protect themselves, that they have the plant cell also have to protect themselves from this zoo. They have a pattern recognition signal. So through this pattern recognition signal, they try to identify the bacterial zoo so that they can initiate the plant defense response through PTI. Recognition of this molecule signature invokes cascade of events. So once this PAMP triggered immunity recognizes the bacterial zoo, they recognize and they invoke several signaling molecules like activation of mitogen activated protein kinase, calcium signaling, callose deposition, phytoalexin production, burst of reactive oxygen species, and transcriptional reprogramming that ultimately led to the PTI. Now, coming to the third line of defense is the R gene, what is known as resistant gene. So this is known as effector triggered immunity. So this senses the effector molecule of the pathogen through cytosolic nucleotide binding site reducing repeat, commonly known as NDS-LLRR, encoded by the resistant gene, and evokes a strong and rapid hypersensitive response in resistant plant with systemic signaling. And by this ways, hypersensitive response that causes the cell death and restrict the pathogen to spread. So coming to the one by one, the first line defense is the plant cell wall. Once the pathogen invades, it comes to the apoplast, where the secondary metabolites actually try to overcome this bacterial pathogen. When it not failed, then the second line of immunity is this PAM-triggered immunity, where there is some molecular signature which recognizes the bacterial effectors and then try to protect the plant from these bacterial effectors. But once this bacteria successfully invade these PTI, then it comes the third defense of line, that is the effector triggered immunity, that is commonly known as resistant gene or R gene. And it was try to initiate the hypersensitive response against this through this ETI and try to make the plant resistant against this pathogen. So this bacteria zoo equipped with different secretion system to carry out the essential secretory function, including other functions like adhesion to the host, horizontal gene transfer through conjugation, etc. There are 6 different types of effector secretion system has been reported in this zoo. That is TS1, TS2, TS3, TS4, TS5, TS6. And TS2 and TS3, these two secretion system were well worked out in plant system in terms of to rise bacterial leaf blight. So what is the work of this secretion system? T2SS is a host cell degradation. It try to degrade the host cell wall, and T3 is to manipulate the host gene to cause the pathogenesis. Similarly, T1 is addition of to the host cell. T4 is the bacterial completion, bacterial conjugation. T5 is the addition to the host cell, and T6 is the microbial competition. So most of the studies, as I said, that secretion system 2 and 3 has been done. And please remember, secretion system 2 is mainly responsible for the degradation of the host cell wall, and secretion system 3 is host cell manipulation, so that the bacterial zoo can easily survive in the host cell. So this is the example of this type 2 secretion system. This type 2 secretion system has 3 enzymes, cell wall degrading enzyme: cellulase, cellulobiosidase, lipase, and esterase. So if you see the wild type, see the disease severity. But once this type 2 secretion system was mutated, there is no disease. So the thing is this: if you mutate the secretion system, then the disease can be hampered. And when this complemented, then again the plant become tolerant. So the type 2 secretion system is very important to control the bacteria from infection. So now coming to type 3 secretion system, as I said, there are 6 secretion system of this bacteria. So basically this secretion system helps them to transport virulence protein and enzymes to disrupt host signaling and hijack the host metabolism for their own growth and development. So this bacteria try to survive by using the host machinery. the rice host machinery so that they can multiply. And for that, they use the host machinery system. So T3 secretion system is main virulence determinant of the zoo and is encoded by conserved HRP and HRC locus, hypersensitive pathogenesis related locus. So this protein secreted by the T3SS are known as effector protein, which is either transcriptional activator like TAL-like effector or non-TAL-like effector. So these T3SS are commonly known as effector protein of the pathogen, and they are divided in two classes. One is the transcription activator like effector, and another is the non-transcription effector like protein. This TAL, the transcription activator like protein, proliferates the zoo and establishment of infection in the host plant by altering the host transcription machinery through upregulation of selected host gene required for multiplication of the pathogen. So as I said that once this bacteria enters into the plant cell and it hijacks the host transcription machinery to propagate themselves in the host cell. And what is non-TAL? Non-TAL promotes virulence through suppression of the host innate immunity. So there is a difference between the TAL and non-TAL. So there is several examples of TAL and non-TAL. Mostly TAL has been reported in the literature. Very few non-TAL effectors has been reported. So these TAL effectors and the cognate effectors genes. So AVR, avulance factor XA10. This is a TAL effector, and XA10 is the cognate effector. So this is the protein. AVR XA10 is the protein, and this is the gene XA10. And similarly, AVR XA23, XA23, AVR XA27, 27 have been cloned, and virulence function of other TAL effector, these have been studied. So these are the few effector TAL effector, they have been widely studied. The PHXO1 TAL effector pursued the expression of host susceptibility gene OS8N3. This is commonly known as nodulin 3 gene family, and this is basically recently renamed as OS Oryza sativa sweet 11. Sweet 11 is a basically transporter. So the bacteria use this host machinery transporter to migrate from one cell to the another cell. So in the Japanese cultivar Nippon bear, it is activates the virulence by inducing this sweet 11 gene. So I will show you how the people has been used this sweet 11 gene. So these are several examples. So the take-home message from this is this: if you mutate the effector binding element of this zoo AVR XA7 effector in the host sweet 14 promoter, we can develop resistance against this zoo. So what people have done, they actually, this is the host gene, sweet 14. As I said, this is a transporter gene, and it is present in the rice plant, OS11N3, now known as sweet 14, or sweet 11 and sweet 23. These are the three well sweet protein has been characterized. So, and this bacteria binds to the promoter of this sweet 14 gene in the effector binding element. If we mutate the effector binding element of the sweet gene, a promoter region, which the effector binding element is present, if we mutate this effector binding element, this AVR XA7 effector cannot bind, and by this way the bacteria cannot propagate. So this way one can develop this disease resistance against this zoo. Similar deletion of this effector binding element of XA7 in wild rice confers broad spectrum resistant to bacterial disease. So here for XA7, they have done for different strain of X zoo, and they have developed a broad spectrum resistant. So these are the example of the rice R gene, cloned R gene, and the cognate AVR genes. So say for example, XA21. This is, as I said, this is the resistant gene, LRRLK, and the cognate AVR is the RAXX, and the encoding protein is unknown. There are several R genes has been in rice has been cloned. So sweet 11 is XA13, sweet 13 is XA25, and sweet 14 is XF41. These are the resistant gene, R gene of the host rice plant, and these are the encoded protein, and these are the cognate aviar, the bacterial virulence effector protein. So the how this XA21 mediated resistance has been developed, as I said, that XA21 is a R gene. So this is a kind of LLR RLK gene originated from the wild rice species, Oryza longis terminata. You know that all the wild species are resistant, but all the cultivated are susceptible, because the wild species, they have very small size grain, and the yield is less. But due to domestication and of need of the hour, so we have domesticated the wild rice to the cultivated rice, and by this way, the most of the treasure gene or disease resistant gene has been present in all the wild rice. And so people has cloned this XA21. This is R gene, resistant gene from wild rice, Oryza longis terminata. This is the first rice gene, R gene, has been cloned from this Oryza longis terminata. So XA21 have been proved to confer broad spectrum resistance against this zoo. So how it works? Basically, as I said, that XA21 is mainly regulated by phosphorylation. Okay, so XB24, it phosphorylate and activates the XA21. But during this zoo infection, the XB24 dissociated from XB21, following which the XA21 is dephosphorylated and provide disease resistance. So the thing is this phosphorylation and dysphosphorylation. When this R gene XA21 is work with the XB24, XB24 phosphorylated, and then it activate this XA21. But when this bacteria infect, the zoo infect, the XB24, this is basically dissociate from the XA21 in the presence of bacterial, the zoo infection, and followed by the XB21 is dephosphorylate, and for that reason it acts as a defense response, and the bacteria cannot progress disease and provide the disease resistance by the activation of XB21 and inhibition of XB10, which is present in the nucleus. So there are the several different other methods than for tackling this disease. As I said, that these are the different R genes, as already reported. So people have also used marker-assisted breeding to develop this bacterial blight resistant rice plant. And this is the variety, high-yielding variety, as like Swarna, Samba Musuri, Pusa Basmati. So they are heavily infected with this bacterial leaf blight, but through breeding, molecular breeding, scientists has developed bacterial leaf blight resistant rice plant, and these are the reference for this work for using the different resistant gene. So one of the classical work developed by Dr. Noves V. Swanti when he was in CCMB Hyderabad in collaboration with Indian Rice Research Institute in Hyderabad, Sundaram, Dr. Sundaram. So what they did, they introduced this disease, the bacterial leaf blight gene from one ERI variety, International Rice Research Association in Philippines. They have developed one bacterial leaf blight gene, and by this use as a donor plant, which is always male, they crossed with the PR106 is the recurrent parent, a female, and the F1 has developed, and then again crossed with the recurrent parent. Two round of back crossing and 3 round of selfing, they develop SS1113 variety. Okay, this is bacterial leaf blight resistant variety by introducing the IRBB62, this line in the Indian cultivar. Now the Samba Masuri is a high-yielding variety, very popular in southern part of India. So, but highly infected with this bacterial leaf blight. So, what these two group, Dr. Sonti and Sundaram, they have done, they use this SS113 as a donor, and they introduce this bacterial leaf blight gene from this ED variety SS113 to the Indian cultivar Samba Musuri as a act as a recurrent parent. They cross it, and then again they cross it with this backcross with this Samba Musuri, again backcross with this Samba Musuri. So this is the basically the donor, and the Samba Musuri is the recipient. So but through successive three round of backcrossing and then selfing into 2002, they developed this bacterial leaf blight resistant gene, the resistant rice plant of Samba Musuri, improved Samba Musuri, which possesses XA5, XA13, and XA21 gene, resistant gene from these three different sources, and by this way they are protected from this bacterial leaf blight disease. So what they did, they have first identified the tightly linked markers, molecular markers for these three independent three R gene, XA21, XA13, and XA5. And during this crossing, they used as a this marker to select the positive plants. So you can see here there are 145 plants they have screened, and out of the 145, the red star one, the positive plant, which consists of XA21 gene in these rice lines. So out of 145, 44 plants are positive to XA21. Similarly, for 44 plants, 23 plants are positive for XA13 R gene. Similarly, for 23 plants, 11 plants are positive for XA13. So in a single backcross, two round of backcross and three round of selfing, they developed this improved Sambar Musuri by introducing this XA21, XA13, and XA5 in a single high-yielding rice cultivar Samba Musuri to make a broad spectrum resistant against the bacterial leaf blight. So this is a great success story what our scientist has done for the developing these three, by introducing the three bacterial blight resistant gene in Samba Musuri high-yielding variety. And this is the outcome you can see here. This is the improved Samba Musuri variety in the field. As I said, that this is done by Dr. Swanti and that Dr. Sundaram from IRRR. And you can see here the improved Samba Musuri and the Samba Musuri, they are almost same in taste, in color, and yield. So there is nothing lost has been made during the cross of introducing the three bacterial blight resistant gene. Now coming to genome engineering approach, this is very important. Okay, so there are different types of genetic engineering, either by knockout or knockdown or overexpression. Okay, so when we do knockout we do for the susceptibility genes, recessive resistant allele, and protein kinase. For activation, we use the defense signaling gene, antimicrobial peptide, resistant protein, and pathogen associated molecular patterns. So these are the candidate genes which you use for engineering. And for overexpression, we use this hypersensitive gene, receptor gene, and antimicrobial peptides. And through tissue culture, we develop this transgenic edited plant, and then we finally develop a resistant transgenic plant through the either genome editing or through overexpression. But knockout using genome editing technique is gaining importance due to concern associated with the transgenics, due to genetic regulatory rules actually. So nowadays the genome editing is one of the important platform to edit any gene to develop the disease-resistant plant. So there are several examples you can see here has been time to time through this transgenic rice overexpression. Okay, like this first transgenic rice line containing XA21. This is the R gene. In type E309 was developed. This is the one Japonica rice developed by overexpressing XA21 in this variety, rice variety, and that bacterial leaf blight resistant line has been developed. Similarly, for IR72, MS63, and IR51500, these are three high-yielding rice variety. They are has been developed through for bacterial leaf blight by overexpressing this XA21 gene. Okay. So XA21 is one of the major candidate gene, resistant gene, has been utilized extensively to develop the bacterial leaf blight. Okay, hence conventional breeding combined with marker state breeding or genomics assisted breeding is the preferred strategy for developing resistant lines or variety to control bacterial leaf blight. Okay, so before that I have shown you how this Samba Missouri has been introduced the 3XA gene and develop an improved Samba Missouri by breeding by overexpression of XA21 gene in different high yielding rice cultivar. People also develop the resistance, but due to this regulatory issue, it has not been seen in the rice field. But genetic engineering is genome editing is one of the steps that government has really actually allowed the release of genetic genome edited lines for cultivation and release in the farmers field. So, what is genome editing? It was one of the promising genome engineering technology, including zinc finger nuclease (ZFNs). There are different way you can do ZFNs, TAL effector nuclease, and CRISPR-Cas9. Okay, which is revolutionize the biology by enabling the targeted modification of the genome. So you can use any of the technique to edit any of the gene so that you can develop the desired plant. So, as I said, that the effector binding element, if we eliminate this in the promoter of the susceptible gene, or if we add this effector binding element to promote promoters of executor of resistant gene through genome editing, could enhance the resistance to bacterial leaf blight. So, I will show you some example. So, example is one of the great example is the OS sweet 14 genes. So as I said, the bacterial this ABR's XA and PTS XO3, the bacterial effectors that binds to the promoter of the host gene, OS sweet 14, to take advantage to propagate within the plant cell. But if we engineer the promoter of OS sweet 14 gene of rice, where this effector binds, then we can stop the binding of these effectors to the promoter of this host gene, so that the bacteria cannot propagate, and this can be done through this talent or CRISPR-Cas9 thing. So this is the host gene. You can see here OS11N3. This is the sweet 14 promoter. Okay, and you can see here, and this bacterial effector ABRXA PTHXO. So in the Tata box region, the single mutation has caused in the effector binding element of the host gene, so that the bacterial effector cannot bind to the host gene, so they cannot propagate. So this is the wild type, the normal plant, and this is the edited line. So you can see here the bacterial infection was nothing actually. When the effector binding element of the host gene has been edited, so that the viral effector cannot bind to the host gene, so that the plant become tolerant. So mutated lines showed strong resistance to both AVR-XA and PTH-XO3 dependent on the exo strain. This was a landmark work, has been published in Nature Biotechnology and done by Lee et al. In 2012. Another work in Nature Biotechnology done by Zhu et al. 2019. Similarly, they have done that they have edited the effector binding element in the promoter of sweet 11, sweet 13, and sweet 14 by CRISPR-Cas9. The previous one I have shown you by this TALEN approach. TALEN is the TAL effector nuclease method, and by Lee et al. And this was by Zhu et al. By CRISPR-Cas9 technology. And rice lines construct broad spectrum resistant to Zhu were created. So this Kitake is the wild type. Okay, and these are the different bacterial pathogen. Okay, strain. So you see here in the wild type, the disease infestation, but in the edited light, there is no, almost nil disease infestation. So by this way, the phenotype of the disease reaction in the wild type Kitake and the gene edited lines MS14K after inoculation with zoo strain 1, 2, 3, 4, 5. There are 5 strain has been used for this checking of this broad spectrum resistant against this sweet 11, 13, and 14. Similarly, Olive et al. 2019 edited a sweet 11, 13, and 14 for Jew resistance. So they have edited the also similarly the sweet 11 gene, and you can see here the wild type, the disease infestation. But in the edited line CS1A, very less, nothing was shown. Even in CS4A and CS6C, nothing. So the thing is this: what they have done, the thing is they have done that the promoter of the sweet gene, where the effector binding element is present, they mutated the effector binding element of the host sweet gene, so that the bacterial pathogen effector cannot bind and propagate. So by this way, simply mutating the effector binding element of the host gene prevent the pathogen to sit and to do the transcription and translation. So what is the conclusion of this whole study? The rice xanthomonas system, properly known as zoo pathosystem, is a powerful model for research towards solution in disease control. Although tremendous progress has been made in the past decade, there are still many queries and challenges has been there because this bacterial pathogen, their strain is mutated, and so they can evade this defense resistant gene. For example, where there are many resistant gene in rice against this bacterial leaf blight or streak, the XA5 confirmed resistance to both zoo and zoo C. This is Oryza cola. Does any other identified XA gene have the same effect? So only XA5 confers broad spectrum resistance against the two the effectors. But is there any other R gene that can be identified to so that it can do the similar kind of work? The partners and or components associated with this R protein remains largely unknown in rice. So as I showed you that the most of the cases I have shown you a tail effector, but there is very few in non-tail effector has been reported in the literature. It would be interesting to understand how R gene activate downstream signaling components and trigger plant defense response system. It is totally unknown till date. TAL effector injected in plant cell have to be translocated into the nucleus to bind to the target susceptible gene for virulence. However, the underlying mechanism needs to be further elucidated. How this translocation from this one cell to the nucleus for this viral pathogenesis has not known till date. Altogether, a comprehensive understanding of the molecular interactions between rice and Xanthomonas oryzae is the pivotal for more efficient and durable disease control. So that's all from my side. Thank you very much. I will be happy to answer any of your questions. Sriram.
Yeah. Sriram. Okay, so let me take care. Thank you, Manoj, Dr. Prasad, for excellent talk. I think the students got enriched, and not only students, we also got enriched by your talk. We learned so many things. Now this floor is open for any question answer, so feel free. And the students and everybody else present here, if you have any question, please ask Dr. Prasad.
Hello, sir.
Yes.
Sir, I have one question. Are all the TAL effectors, whether are these the membrane bound or these are soluble proteins?
So mostly they are membrane bound.
Okay. So they are released after some time. What is the mechanism happening?
That is still not known actually. How the TAL actually work, it is not known.
Okay, sir.
So that's in the last slide I have given more unanswered question that to be answered by the scientist.
All right, sir.
Any more questions?
Yes.
Can you hear me now?
Yes, Sriram.
Okay. Sorry, actually I had some problem with the mic. Yeah, any more questions, students?
Sir,
Balaram,
I have
So I have a
oh,
question.
Yeah, please go ahead.
Uh, so you like explained like naturally hydrothods can also serve as the entry point for these bacteria. So
yes.
can reducing the number of hydrothods in leaf can serve as a strategy for getting plants more resistant to these bacteria.
Yeah, that thing, but how to reduce the number of hydrothods? But this is the natural process of the plant. That hydrothods are sometimes it acts as a good, sometimes it is bad for the for disease causing.
Okay.
Yeah,
Okay, sir.
Balram.
Balaram Mukhopadhyay had a question.
Yes, I'm from a different department though. I'm a synthetic organic chemist
Mm-hmm.
and working on the field of carbohydrate.
Mm-hmm.
So my interest is that what about the O antigen structure? Because these bacteria, they have a O antigen structure in their surface.
Mm-hmm.
Is that O antigen structure is responsible for the disease progression or the attack?
That in depth I cannot be answered because that in details I have not seen any study in the literature. But as I said, that the bacteria possesses that cell degrading enzymes, so they degrade the cell wall uh, so that it can come to the apoplast and then use the xylem vessel to go to the other parts of the cell. But this your thing, I have no clue how it happens.
Okay,
So
okay.
if Manoj allows me, so I also used to work on bacteria on Vibrio cholerae. So mainly the gram negative, the O antigens are responsible for so many things. And maybe it is degrading enzymes is there, but but somebody can investigate whether
Yeah,
this O antigen has anything to do with the pathogenicity or not.
pathogenicity on it.
Yeah,
Yes.
because the host cell may have some receptor for those oligosaccharides, which
Absolutely.
are allowing the bacteria to just get there.
Yeah. So what we checked for our Vibrio cholerae long back, that these lipopolysaccharide O antigens, and they actually cause all kinds of variations of the polysaccharides on the surface. And they are responsible for initial attachment. Some things are mannose sensitive, some things are fructose sensitive. I think Balaram will be knowing about it. So that's a good area, Manoj. Either you or you can tell somebody to investigate.
Yes, yes, definitely, definitely.
That's a nice question. Yeah.
So Balaram is actually our chemistry professor.
Yeah, yeah, that's why the question angle is I understood. So that is the reason I think transdisciplinary research is required, na? Because
Absolutely.
everybody is different from thinking it from different perspective.
Yes, yes, yes.
So students ask questions.
Students, any questions?
sir, to prevent the infection, you said that some of the scientists engineered the promoter. So
Mhm.
can it affect the normal physiological process of the plant, or how they ensure like which type of engineering they should implement, like how they uh, the first time they thought like we should and how this engineering actually work, like
So look, this sweet gene is a transporter. It requires by the pathogen, whether it is virus or bacteria, to survive on the host cell. And in the promoter of the sweet gene or the host sweet gene, there is a effector binding element site where the zoo basically effector is to go and bind and use the host transcription machinery by fooling the host cell. They use it for their own benefit, the pathogen. So the scientists thought that if we edit this effector binding element site in the host gene of promoter sweet gene, then this bacterial effector cannot bind and they cannot propagate and they cannot use the host machinery to survive on this rice plant. So that is the reason this is very easy to just single nucleotide change, and that cannot allow the bacterial effector to use the host machinery for survival, for propagation. So by this way, we can check the bacterial pathogen for its transcription and translation.
Sir, is this like promoter also work for the host own gene expression? So will not the change in the promoter can affect their normal physiological process?
No, because the thing is this, that the normal cell, it will work, but that is only one in one cell you are doing this thing, so that but if one machinery will be stopped, but other is there, na? So from it will prevent the plant from getting infected by the pathogen. So the suite is one of the transporter. There are several transporters there. Suite either suite 11, 13, 14, 23. Okay, so if we prevent this any of these suite 3 genes, and then it can do not harm the host cell for their any activity. It has been shown there is no yield penalty if you engineer the suite promoter, the effector binding element. It can prevent the plant from getting infected.
Okay, sir. Thank you.
Sir, I have another question.
Yeah, please.
Sir, you talked about using antimicrobial peptides to reduce the zoo pathogenicity. So, sir, how to use them? I mean, how do we deliver it? Like, what is the delivery mechanism of this antimicrobial peptides against zoo?
So that very less research has been done on that aspect, but this is one of the way to counter the pathogen from infection. But how, I cannot give you the answer at present. Actually, but peptide, as people are using, they propose that antimicrobial peptide is one of the way, but how, that I cannot give you rightly the answer.
Okay, sir.
So whatever I
Okay.
have said, they are all documented. That's why I have presented. Otherwise, whatever they are there is no documentation. I didn't say anything.
Okay, I think with
No,
this,
if
yeah,
this pathogen invade the plant or is gone on the surface of the leaf,
The surface of the leaf, not invade.
then peptide can be sprayed. It will be very costly though.
Yeah.
And how much it will retain, we don't know. Because we
yeah,
have a
that,
trend through NGNCs, we have found that in presence of glucose, it gets the biofilm on the any surfaces. But the question is that how much you spray the
Right.
glucose. So in one hand, people do the research to see the potential, but application is a challenging site. So what
Exactly.
question the rest? Yeah.
yeah, that is the main thing. Delivery is the main problem here.
For any disease you
Yeah,
talk
yeah,
about, yeah.
yes.
So any other
Okay,
question?
I think if you don't have any questions, we'll stop here.
So, Sriram, I have
Yeah.
shared these today's talk slides to you, and then
Sure, yeah.
Supratim.
Yes.
Okay.
Okay, thank you.
If the student needs, then you can give them the PDF of the slide.
Sure, yeah.
Sir,
Okay.
A
before
few
leaving,
iGEM
can
students
we have
are
a group?
requesting me that whether they can put your talk in YouTube or not. Still they have this request, I don't know. The iGEM team can tell.
Yes, sir.
So we need his permission then. Yeah.
No, but these are all for education purpose. You can use it.
Oh, great.
Yeah,
great.
yeah,
Thanks
that's
a
fine.
lot.
Yeah.
Thanks a lot.
Thank you.
Students get
Sir,
on
before
very
leaving,
benefit.
can we have a group picture, sir?
Group picture.
Then
Yeah, you
everyone
can take.
open
Everybody
your
has to switch on camera.
Everybody has to switch on the camera. Then, oh, I had very great idea. I had no idea. These kids are very innovative, Yeah. Anyway.
No, I just stop sharing. Then I think everybody will come into the picture.
Yeah. But how to accommodate 27 others? I think who can do that?
No, total 35 I can see.
Yeah, 35. I think some mechanism is there to do. I don't. Chirag, can you help Sriram how to do that?
Yeah, let's give me a second.
Yeah,
Now,
but maybe
I think
Sriram also a
yeah.
young guy. So
Yes, yes.
I'm
All of us are in the view, right?
no. To me, it's
Yeah.
not. But
No?
but you can take, if somebody can see all of us, then can take a snapshot,
Yes.
screenshot or whatever. I don't know. Yeah.
I think all of them are there now.
Sir, I think you can unpin the presentation, and then most of us will be.
No,
The
Sriram,
presentation is.
Sriram, can you
I
see
can
everybody?
see
Yeah, I
I
can
can
see
see many
everybody.
many.
Then go for
Now.
a screenshot. I don't know how to do that.
I can take a screenshot, but everyone has not like switched on their camera. Everyone can switch on the camera, then I can take a
I mean, there's a
screenshot.
maximum that we can have in one field. I think.
No. All can come. Small करके आ जाएंगे.
No,
But
it's a
I don't know how
maximum
to do.
tiles to display, not two.
27.
Okay, tile, spotlight.
I can see 33 people.
So then
I think we have to take maybe two
Yeah.
shots.
I can see 34
Yeah,
people.
34
in
is
one.
good. Yeah. It is something like
Should I
that.
take a? But
Yeah.
everyone has not
Yeah,
switched on the
good.
camera.
Please, all of you switch on the
Everybody
camera.
switch on the camera, please. Yeah.
Should I take now?
Yeah.
Yeah, take.
Take some 2, 3.
Done.
Okay.
There would
thank
be
you.
also share these pictures.
Yeah.
Sure, sir.
Yeah.
Sure, sir.
And on behalf of everybody, Manoj, I'm really grateful and thanks to you. Yeah, because in short notice, he's a person of such stature, and he agreed for the sake of the students. So students you should all thank him. Yeah, he actually did it for the students. Yeah.
I think Subodip also agreed to talk.
Yes, he'll talk on Wednesday.
Subodip is his field actually. Subodip's field.
Yeah. I told Mohit to talk to him. Yeah, so yeah, no problem. Okay.
Thank you so much, sir.
Thank you.
Okay, thank you, sir.
Thank
Thank you very much. Yeah.
Thank you.
Thank you.
Okay.
Thank
Bye.
you. Yeah. Bye.
Okay, Sriram.
Okay, Tapas, I think you're done.
Okay.
Thank
And
you.
the students are still there. They were asking about Arun Lahiri Majumdar. Did they contact?
Okay.
I don't know.
Maybe you can talk to them.
Nobody there. No?
No, nobody's there.
Okay, I'll be back on campus again on Thursday. I'm not doing okay actually.
Okay.
More or less okay, but yeah, okay.
Yeah, maybe you can talk.
No. If Subhajit can talk, then we may not need Arun La. And if they need Arun Lahiri Majumdar, they need to inform me. Okay. Okay then. I'm
Okay.
leaving.
Bye. Okay. Yeah. Thank you.
You stop recording. I will be recorded for hours.
Yeah.